In this lecture, we will examine organisms within the domain Eukarya. The domain Eukarya consists of protists, fungi, plants, and animals. Previous ways of classifying organisms within this domain listed the protists as their own kingdom. However, this classification is considered to be outdated. This is because we now recognize that some protists are closer relatives of plants and animals than they are to other protists. We now recognize distinct protist lineages that have arisen multiple times throughout the evolutionary time. The domain Eukarya consists of organisms with a eukaryotic cell as their base organization. This means that the typical eukaryotic cell has an organization that includes organelles as well as a cytoskeleton. In many eukaryotic cells, you will note the presence of a nucleus, mitochondria, Golgi apparatus, smooth and rough endoplasmic reticulum, as well as features of the cytoskeleton such as microfilaments, microtubules, and intermediate filaments. Take a look at the diagram below to see the typical organization of a eukaryotic cell. We can see the nucleus in the center of the cell that contains the genetic information. This is typically surrounded by the smooth and rough ER. This then extends into the Golgi apparatus. We can also see various types of organelles, such as lysosomes, mitochondria, interspersed throughout. Note that you may also find the presence of chloroplasts as well. Note that the cytoskeleton consists of the microfilaments, intermediate filaments, and microtubules, and the plasma membrane can be extended into microvilli or may have a flagellum attached to it. It is important to note and consider how eukaryotic cells evolved. How did evolution go from the base prokaryotic cell, which is simple in its form, to a complex eukaryotic cell? If you recall the endosymbiotic theory, this plays a very important role in the evolution of eukaryotes. Also recall that there is much structural, biochemical, and genetic evidence that supports this theory. This theory states that both mitochondria as well as plastids are derived from bacterial cells. It is believed that these bacterial cells, the mitochondria and plastids, were engulfed by an early ancestral pre-eukaryotic cell. It's also important to note that all eukaryotes have one organelle in common, that is the mitochondrion. This suggests that the mitochondria evolved prior to the evolution of plastids. If we take a look at the overall structure of the mitochondria, we can see that it is enclosed by both an inner membrane as well as an outer membrane. The inner membrane is considered to be highly folded and forms the inner cristae. Also note that the mitochondria itself contains its own DNA as well as free ribosomes, enabling the mitochondria to replicate independent of the overall eukaryotic cell. Analysis of mitochondrial DNA has yielded important findings regarding the endosymbiotic theory. It is suggested that mitochondria arose from an alpha protobacterium. It also suggests that one common ancestor is present, indicating that the consumption and evolution of a eukaryotic cell and the mitochondrion within a eukaryotic cell occurred only once during evolution. Similar findings have also occurred considering the origin of plastids. However, it is believed that plastids originate from a cyanobacterium. You can see here in the diagram depicted, we have an overview of the endosymbiotic theory, beginning with our pre-ancestral eukaryotic cell consuming a mitochondrion-like bacterium. This mitochondria-like bacterium would then form a symbiotic relationship with the host cell. This then occurred an additional time with a photosynthetic prokaryote, such as a cyanobacterium. This would then allow for the eukaryotic cell to possess a mitochondrion as well as a plastid, and this cell would then be able to carry out photosynthesis. When considering the evolution of plastids, it's believed that plastids evolved from the endosymbiotic process with a photosynthetic cyanobacterium. It is also believed that this evolved in two separate lineages involving both the red algae as well as the green algae. Evidence to support this conclusion derives from the plastid membrane, which closely resembles that of a cyanobacterium. If you also examine the transport proteins that are present in the inner and outer membranes of the plastid, they are also considered to be similar to a cyanobacterium. Furthermore, it is also believed that both red and green algae underwent a secondary endosymbiosis event. This has led to the derived lines of stromatophiles and alveolates in the case of secondary endosymbiosis of red algae, and euglenids and chloracneophytes in the case of green algae. Vestigial nuclei called nucleomorphs is still present within the plastids of chlorarachneophytes. This nucleomorph region still has genes that are transcribed and is very genetically similar to green algae. The diagram below represents the primary and secondary endosymbiotic events. In the primary endosymbiotic event, we have a cyanobacterium that is ingested by a pre-ancestral eukaryotic cells. In this instance, this led to the two different lineages, red algae as well as green algae. These two lineages then underwent a secondary endosymbiosis event, which led to the formation of stromatophiles and alveolates that contain the red algae, and euglenids and chloracneophytes that contain the green algae. Within the domain Eukarya, current phylogenetic analysis has identified four main supergroups. These include Excavata, SAR, Archaeplastida, and Uniconta. It is currently unknown what the common ancestor of these four supergroups is. Thus, they are commonly shown diverging from one node. In addition, it is also unknown which group originated first.